All right, so yesterday we labeled the plant cell and we said this won't take nearly as long because pretty much all the structures are the same, okay? And if they're the same, they do the same thing. So what's the big purple thing? Okay, so that's the nucleus. Oh, I don't want the big black marker though. I like the small black marker. That's better. Okay, and then I added a few labels on, but I don't remember exactly which way I made them go. So I'm just going to draw the one that's from this darker purple thing over here. What is it called? Nucleolus. Okay, what does the nucleus do versus the nucleolus?
The nucleus kind of just protects the nucleolus, and then no.
The nucleus contains all the DNA, and the nucleolus copies the DNA.
Right, yeah. Okay, the nucleus stores the DNA. The nucleolus copies it, or uses those makes those copies to help with protein synthesis. It also makes ribosomes. If I was just on a test asking for the function of the nucleolus, any one of those things would be good enough for the answer. Okay? All right, and then what's the outer layer called? Nuclear membrane. Remember that membrane is special in that nothing gets in or out of it except through the pores, and the only thing that gets through the pores is genetic material. Okay. All right. What did we call the fluid that fills up the cell?
Cytoplasm.
Cytoplasm. Okay. What was its function? I would say that's somewhat true, but the kind of high school level answer that I would want, it facilitates diffusion. I would also accept assists with cell transport or intracellular transport or something along those lines. Okay, in as much as it holds the organelles up, it's not really its function, it just kind of does that. Okay, its more important function is to allow materials to diffuse throughout the cell. Okay, all right, the two yellow gear-shaped deals, those were not in the plant cell. They are unique to an animal cell, but we didn't talk about them before when we were talking about the modes of nutrition. So they're only present in an animal cell. Okay? Now, animal cells can change shape, yeah?
Yes.
Right? But a plant cell really doesn't, right? It's got the rigid cell wall that holds its shape. So the issue faced by animal cells is that during cell division, they could be stretched or twisted or compressed or something like that, and that could result in the genetic material not getting where it's supposed to go. Okay, so these things here, which are called centrioles, ensure that during cell division, the chromosomes, the DNA, get where they're supposed to go. So it works something like this. Okay, that's an animal cell. During cell division, okay, in fact, this is true for any cell. During cell division, there's a point in cell division called metaphase. You don't need to know that, okay? Not till grade 11, right? So in metaphase, all of your chromosomes actually look like pictures of chromosomes that you may have seen, okay? And they all line up across the middle, what's called the metaphase plate, okay? And these two centrioles migrate one to each pole. Okay, so you have a centriole at the top and a centriole at the bottom. And then what they do is they send out little fibers. And these fibers will latch on to the copy of that chromosome that is supposed to go to each side. Okay, I'm not going to draw them all, but you get the idea. Okay, so they latch onto these things like a cable, and then right before the cell splits in half, the centrioles kind of reel them in, right? They pull them apart off the metaphase plate and make sure that the right number of chromosomes get to each side, and then the cell will actually divide, and then you'll have all the chromosomes necessary. The nucleus reforms around them, okay, and your cell is off, and then the centriole will replicate itself so that there are again two in each cell. Right? So when it comes right down to it, I don't want an explanation that detailed. I'm just explaining to you that's how they work. Okay, if you were telling me what's the function of a centriole on a quiz or a test, helps with cell division would be good enough. Okay, Pietro.
How about Down syndrome, like people like that, like an extra chromosome?
Yeah.
It's like a mistake by the controls, or is it like, person was already had an extra?
Usually it isn't I mean, it could be a problem with the centers. There's a lot of things that could cause one cell to get more DNA than the other. It could be a problem with the centrioles, like maybe the chromosome just didn't pull apart properly when they pulled. It could be a problem with replication. Okay, so that would be long before the centrioles ever even went to the poles. There could be a problem with replication, and it could have been done more than it should have. Any number of things can cause that, but it has to happen when the gamete forms. Okay, so if you have a cell that has a mess up, okay, during this part, you're never gonna know that. It's not gonna cause anything bad to happen to you, except for those two cells are probably gonna die, okay, which you'll never know, right? But if this happens as an egg or a sperm is being formed, that's a big deal, right? Because that's the only copy of DNA that organism's gonna get, and it's gonna replicate that over and over and over again. Does that make more sense? Yeah. Okay, so that's the role of centrioles, okay? They're pretty important, but they're only present in animal cells because a plant cell doesn't need these. It's not gonna change shape, twist, or stretch or something during cell division and have the risk of losing some of its DNA or having it end up in the wrong cell, okay? So plant cells don't need them, thus they don't have them. They're only present in animal cells. Okay, what's the brown stuff with the turquoise dots?
Rough ER.
Rough ER. Okay, what does the rough ER do again?
Protect the nucleus.
Nope.
Oh, no.
Isn't it like kind of the shipping and create like?
There is
taking out
something in there that
ribosomes?
does that, but it isn't the rough ER. The rough ER transports proteins, okay? Because the little dots, the ribosomes, do what? They make the protein, yeah, okay? So they're covered in things that make protein. It's logical that they would transport the protein that's being made all over the place. Okay, so the little turquoise dots then, okay, same as in the plant cell, those are ribosomes, and their function is to make protein. All right. All right, what are the blue sausage-shaped things?
Yeah.
Mitochondria, right. Okay, so the mitochondria do what?
Assist in cell respiration.
Right. Okay, they carry out cellular respiration. Right? That is what the glucose is converted into energy. Okay. All right. We got two of these little bubbles, and it's kind of hard to tell them apart. All right, so here's what we're going to do. We're going to write that this one has food in it. Okay, so if there's food in it and digestion is occurring, this is a what? Lysosome. Digestion is occurring. If it just had food in it, it would be a vacuole. Okay, so this would be a lysosome. All right, this one here, no food, just the enzymes. Okay, and I think I only mentioned this word once, so I'll just write it in. This is a peroxisome. Okay, that's the thing that actually comes off of the Golgi apparatus. Okay, it's just a vesicle that has the digestive enzymes. It's not a lysosome until that merges with a food vacuole and digestion begins. Okay? Okay, what's the outside of the cell? Cell membrane. Okay. And if I asked you for a function of the cell membrane, what two words would you write for me? I only mentioned them once, so I just wondered if anyone actually wrote them down. Endocytosis and exocytosis. That is movement of materials in and out of the cell. Endocytosis endo meaning in, cytosis meaning cell, okay, into the cell. Exocytosis, exo means out, cytosis means cell, out of the cell. Okay.
What's the one out of the cell?
Exo. Okay, what's the yellow thing? Golgi apparatus, right. Okay, and the Golgi apparatus prepares materials for removal from the cell. Okay, we could also say produces peroxisomes, but the better thing would be that it prepares stuff for removal from the cell. Okay, that's either wastes or it's materials that are produced by the cell and need to be secreted. Okay, it essentially prepares things to be removed, or for exocytosis, prepares them for that. All right, we don't have anything on the smooth ER. Okay, so I think I drew a line down for this, or did I draw it over there? Anyway, that is the smooth ER. What does the smooth ER do? Oh. Lots and lots of things. Okay, the smooth ER, number one, detoxifies poisons. Okay, so if you consumed anything, okay, or breathed in anything, and it got into your bloodstream that was toxic, okay, your smooth ER would be the parts of the liver cells that would take care of that. Okay, they're responsible for detoxifying poisons. They can also help to break down complex carbohydrates. Okay, so if you had, a big plate of spaghetti and you ate all of that starch, okay, the smooth ER could help to break down the complex carbohydrates. It's not really digestion, okay, that's the job of the lysosome, but it can break down complex carbohydrates. Okay, it can also make lipids, hugely important function. Every membrane in your cell, the cell membrane, the membrane of every organelle, they're all made of the same stuff, fat. Okay, and the smooth ER makes fats, makes lipids. Okay, so it produces lipids. Lipids are responsible for repairing membranes. Okay, so I had detoxified poison, break down complex carbohydrates, makes lipids. Oh, and fat soluble hormones. Okay, it can produce some fat soluble hormones. Okay, so if your cell didn't have any smooth ER, it would be in real trouble. Okay, because that stuff does a lot of things, and every one of them is really important. Okay. All right, any lines on there we didn't get to? Okay, I think that's all of them, right? Okay, so big thing here for the cells, guys. On Monday, after we go over today's quiz, we're going to go over in detail what each part of the cell does and how it does it. Okay, and then we'll move into cellular transport. Maybe not Monday, but maybe Monday, and then most of Tuesday will be on cellular transport. Okay, we'll be in the lab next week doing our cell anatomy lab. And then we'll have a few other activities during the week that we'll be working on. Okay, the first half of the unit goes pretty fast. Like, we'll be done it next week. Okay, the first half of the unit is on the cell. You don't have to spend a lot of time on that. Cells are small. Okay, we just need to know what they do and how they do it, what they're made of. Okay, and that's really about it. All right, so next Friday's quiz will be cell diagrams and you labeling and telling me what the parts do. Okay? All right, so I do not have the Chromebooks today, but my intention was to give you about the last 15 minutes or so here to work on your lab reports. Okay, your microscopy lab reports. So you've got a little bit of time there if you want to run some ideas by group mates, okay, and just talk to them about, what you're writing for analysis, okay. If you got your own machine, then maybe you can work on the lab drawings or something like that, okay. But you got a little bit of time here to work on that lab report.